Imagine you're staring at this massively detailed map of the world. Like, you can trace the sharp ridges of mountain ranges, you can follow the winding paths of rivers, and clearly see all the distinct borders of the continents.
Right, a highly detailed topological map.
Exactly. It's a beautiful, high-resolution rendering of geography. But there's this massive catch. Every single label on this map, like the names of the countries, the oceans, the cities, it's all written in a completely alien language.
Oh, wow. Yeah.
So, there are boundaries. You can literally see the differences in the terrain, but you have absolutely no idea what any of it actually means. You can't navigate it.
And that analogy, honestly, that perfectly captures the current frustration in the whole field of single-cell biology right now.
Really, just that sense of being totally lost despite having the data.
Yeah, exactly. Because artificial intelligence has become, incredibly powerful at drawing the map, but it completely abandons us when it comes to actually reading the labels.
And that is exactly the mission for our deep dive today. We're looking at this fascinating technical report published in Nature Cell Biology. It's about a new machine learning framework called expiMap.
Right, which stands for Explainable Programmable Mapper.
Right, and the goal here is to figure out how scientists are, how they're finally prying open the black box of AI, taking deep learning from this tool that just blindly sorts data and turning it into a system that actually explains the underlying biology back to us.
Which is a huge paradigm shift.
It really is. But before we get into the heavy math of how expiMap breaks that black box, I think we need to understand what this map actually is. We hear the term single-cell atlas a lot in this research. What are we actually mapping here?
So basically, we are mapping healthy human biology at the absolute highest possible resolution. Over the last several years, RNA sequencing technology has advanced so much that we can now look at a tissue sample and literally sequence the RNA of individual singular cells.
And we're talking about huge numbers of cells, right?
Oh, yeah. Millions of them at a time. And by analyzing which specific genes are turned on or off in each individual cell, we build these atlases. They act as these massive coordinate systems of what a normal, healthy cellular ecosystem is supposed to look like.
Okay, so we have a baseline, like a giant, healthy reference map.
Exactly.
And the practical application of this is something called reference mapping. So you take a new query data set. Let's say it's a blood sample from a patient who is severely ill, and you want to overlay those 6 cells onto your healthy atlas to just see where things deviate, to see what went wrong.
Right, but the real challenge is doing that overlay accurately, because when you sequence a new patient's cells, you inevitably introduce what we call batch effects.
Batch effects. Is that like technical noise?
Yeah, it's pure technical noise.
Yeah.
Maybe the patient sample was processed in a different lab or on a different sequencing machine, or honestly, maybe it was just processed on a Tuesday instead of a Wednesday, and the temperature in the room was slightly different.
Wow, it's that sensitive.
It's extremely. And current deep learning models like SCVI, for example, they're fantastic at scrubbing away that technical noise. They can seamlessly integrate those new cells into the reference map. But, and here's the catch, to achieve that seamless integration, the AI compresses all those thousands of genes into what we call a latent space.
Okay, I really want to pause on this idea of a latent space, because this concept seems to be the entire root of the black box problem we're talking about.
It absolutely is.
I always think of it like a complex fruit smoothie. Like if someone hands you a smoothie made of 20 different ingredients, your brain doesn't process it by identifying the exact molecular weight of the strawberries versus the kale, right?
Your brain just doesn't work that way.
Exactly. Your brain creates an abstract latent space of flavor profiles. It's sweet. It's a bit earthy, it's tart. You've basically compressed 20 raw data points into 3 abstract feelings.
That's a great way to visualize it. It's a process of extreme dimensional reduction. A single cell might express 20,000 different genes.
20,000.
Yeah. And an AI simply cannot efficiently map millions of cells while tracking 20,000 independent variables for every single one. So it compresses those 20,000 genes into maybe 20 or 30 mathematical dimensions.
The smoothie flavor profiles.
Exactly. Those dimensions capture the overall state or flavor of the cell. And we can look at this compressed space and see that, yes, the AI successfully separated the healthy cells from the sick cells.
But the problem is,
if we ask the AI, hey, which specific biological pathway caused this sick cell to shift to the left on the map, the AI cannot answer. It used highly abstract math to draw that boundary, not biological concepts.
Which makes the model computationally brilliant, but, practically useless if you're a researcher trying to design a targeted drug. You can't design a pill to target abstract mathematical dimension number 4.
No, you definitely cannot.
You need a gene pathway. And I guess this is exactly where expiMap steps in. Because the paper claims this system marries the raw power of deep neural networks with actual human biological knowledge.
Yeah, and to understand the mechanics of how it actually pulls that off, we really need to look at its architecture. expiMap uses what's called a conditional variational autoencoder.
Okay, let's break that down. I get the basic autoencoder part. It's sort of shaped like an hourglass, right?
Right, exactly like an hourglass.
So the top half, the encoder, takes the massive 20,000 gene input and essentially squeezes it down through a bottleneck into that compressed latent space. And then the bottom half, the decoder, tries to reconstruct the original 20,000 gene data from that tiny compressed summary.
You nailed it. That's the basic autoencoder.
But what does the conditional variational part actually mean in this specific context?
So conditional just means we're feeding the model extra context alongside the raw gene data. We condition the model on the batch information.
Ah, so we're explicitly telling it, hey, this data came from lab A and this came from lab B.
Exactly. We tell it which lab it came from or which patient. And this allows the model to isolate and systematically subtract that technical noise, those batch effects we talked about earlier.
Okay, that makes sense.
But the real magic of expiMap, the thing that makes it explainable, lies in how it structures that hourglass shape. The top half, the encoder, is completely nonlinear.
Meaning it's highly flexible.
Highly, highly flexible. In a nonlinear neural network, variables are multiplied and transformed in these incredibly complex web-like cascades.
Like a giant tangled knot of math.
Yeah, a tangled knot. And this flexibility is exactly what allows the top half of the hourglass to capture and untangle all those messy, complicated batch effects. But, and this is crucial, the bottom half of the hourglass, the decoder, is strictly linear.
Okay, stop right there. Why is a linear decoder the secret ingredient here? Why does just making a straight line suddenly make an AI interpretable?
Think about that tangled knot. In a nonlinear equation, if you change just one single input, it triggers a cascade of compounding changes throughout the whole network,
Right.
making it mathematically impossible to trace the final output back to one single starting variable. But in a linear model, variables are kept mathematically isolated. The final output is literally just a simple weighted sum.
Oh, I see. So there's no tangled web.
No web at all. If a connection in a linear decoder has a high weight, you know with absolute mathematical certainty that it directly drove that specific output. Linearity guarantees that we can trace the math backward.
Okay, I get the math part.
Yeah.
But a linear decoder alone doesn't solve the alien map problem we talked about at the start. It just makes the math traceable. It's still just math.
Right. You need the labels.
Yeah. And the researchers took it a step further with something called architecture programming. They literally wire the artificial brain using human biology.
Mm-hmm.
The paper mentions utilizing databases like Reactome and PangloDB. What exactly are those, and how do they fit into the wiring of the AI?
So Reactome and PangloDB, these are massive, highly curated databases assembled by thousands of human scientists over decades.
It's like biological encyclopedias.
Exactly. Encyclopedias of biological pathways. They tell us, for example, that out of 20,000 genes, these specific 50 are responsible for the immune response, and these other 30 handle metabolism.
Okay.
The expiMap researchers basically took those encyclopedias and converted them into a gene program matrix, a GP matrix.
So it's basically a binary mask, like a massive spreadsheet of ones and zeros.
Yes, precisely a binary mask.
So if row 1 is the immune response, every single gene involved in immunity gets a 1, and the other 19,950 genes get a 0.
You've got it. And the AI uses that spreadsheet as a literal mask over the network's weights. Remember that bottleneck layer, the latent space?
The smoothie. Yeah.
Right. expiMap forces each individual node, each abstract dimension in that bottleneck to correspond to one specific row in the spreadsheet. Node number 1 is explicitly hardwired, so it can only communicate with the genes marked with a 1 in the immune response program. Node number 2 is hardwired only to metabolism genes.
Oh, wow. So the latent space is no longer just abstract tartness or sweetness.
Right.
It actually has real biological labels.
Exactly. The AI is literally forced to compress the data using our human concepts.
Okay, but I have to play devil's advocate here for a second. If we force the AI to only look through the lens of Reactome and PangloDB, aren't we kind of crippling it?
How do you mean?
Well, we know those human databases are incomplete, right? We discover new gene functions like every single year. If we lock the AI into this rigid binary mask of only what we already know, Aren't we just building a multi-million-dollar echo chamber? Like, it will never discover anything new if we only let it see the old stuff.
That is a brilliant point, and honestly, it's a critical flaw in a lot of older knowledge-based models. They are way too rigid. But expiMap circumvents that echo chamber effect by transitioning from what we call hard membership to soft membership.
Soft membership.
Yeah. It treats the GP matrix as a very strong suggestion, but not an absolute law. While the weights for genes that aren't in the official program are initially fixed to 0, expiMap introduces this clever mathematical mechanism called L1 sparsity regularization.
I've actually seen L1 regularization mentioned in other deep learning papers, but help me visualize how it actually loosens up this rigid mask in this context.
Okay, think of the AI as a project manager operating under a ruthlessly strict corporate budget.
Okay, I like that.
The AI's job is to reconstruct the original cell perfectly, using only the specific genes we assign to that pathway. But what if our human database is missing a crucial gene, and the AI keeps failing to reconstruct the cell accurately because it's missing that piece?
Right, because our encyclopedia was incomplete.
Exactly. The L1 regularization acts as an emergency budget. The AI is allowed to pull in a brand new unlisted gene to help, but doing so incurs a massive financial penalty. It's a heavy mathematical tax applied to the model's loss function.
So the L1 penalty mathematically forces sparsity. The AI won't just pull in every single gene willy-nilly because it literally can't afford to pay the tax.
Precisely. It will only pay that heavy tax to add a new connection if that specific new gene drastically improves the accuracy of the final reconstruction.
So it only adds weights that provide a massive return on investment.
Exactly. And because of this mechanism, expiMap isn't just using human ontologies, it's actively editing them. It actually finds new genes that belong to old pathways, refining our databases based on raw data rather than just relying on human assumptions.
That is incredibly elegant. So that solves the problem of incomplete databases. But what about the exact opposite problem? What if our databases are bloated?
Bloated.
Yeah, like a human encyclopedia might have 500 different pathways just for brain function. But if we are mapping liver tissue, all those brain pathways are just dead weight. How does the model avoid getting completely bogged down by irrelevant biological noise?
That's where the researchers implement a second mechanism, which is called group lasso regularization. And it functions sort of as an attention mechanism in the latent space.
Okay, so the L1 penalty manages the individual genes, while the group lasso manages entire pathways.
Exactly right. Group lasso looks at the overall utility of an entire latent node. If a whole gene program, say a brain function pathway in a liver cell, is just not actively contributing to minimizing the model's error, the group lasso applies a penalty that totally deactivates that entire node.
It just shuts the whole thing down.
Yep. It drives all the weights for that entire program to 0. It prunes the dead weight, ensuring the model stays highly efficient and focused strictly on the biology that is actively driving variations in the specific tissue being analyzed.
Wow. Okay, let's take a quick breather and lock in these concepts because we just covered the entire foundational architecture of this model. For you listening, here is your 5-minute recap of how we build a glass box AI.
Let's hear it.
Okay, first we tackle the black box. Traditional latent spaces compress data into unreadable math. expiMap fixes this with a conditional variational autoencoder using a nonlinear encoder for flexibility and a linear decoder so we can explicitly trace the math backward.
Spot on.
Second, the GP matrix. We convert human biological databases into a binary mask of ones and zeros, forcing the AI to categorize data into known biological pathways. Third, soft membership via L1 regularization. The AI has a strict budget, but it can pay a mathematical tax to pull new, undiscovered genes into known pathways, actively updating human knowledge.
Which is huge.
And fourth, group lasso. The model ruthlessly prunes away redundant pathways, keeping the map clean and relevant to the specific tissue.
Perfect summary.
So we've built this dynamic, self-editing, biologically labeled map of healthy tissue. But I want to pose a question to you, the listener, to think about. What happens to this pristine map when a totally novel disease like a brand new virus that doesn't exist in any of our databases enters the chat? How does a model trained purely on known biology process the completely unknown?
And this is the perfect transition to what we call the query phase. We take our trained reference atlas and we introduce that new, sick data set. To process biological variations that the model has quite literally never seen before, expiMap performs something called architectural surgery.
Architectural surgery? That sounds intense. Are we literally cutting open the neural network?
Conceptually, yes, we kind of are. It's a highly specialized form of transfer learning. First, we take the original reference model and we freeze the weights of all those established gene programs. We just lock them down entirely.
Okay.
By doing this, we create an information bottleneck. The model is now highly constrained in how it can process any new incoming data.
I see. So if the incoming disease has a unique novel signature, the lock pathways are going to really struggle to reconstruct the cell because they just don't have the biological vocabulary for the new virus.
Exactly, which is why the surgery part involves grafting new nodes, new latent dimensions right into that bottleneck layer, and these new nodes have fully trainable, unfrozen weights.
Oh, I get it. Because the established nodes are locked and struggling to explain the new disease, the mathematical pressure forces these new unfrozen nodes to learn the novel, unaccounted for variations in the query data.
You've got it. And the paper mentions these new nodes can learn what they call de novo programs, meaning they learn entirely unconstrained biological circuits completely from scratch without any predefined spreadsheet of genes telling them what to look for.
So they are discovering pure, novel biology.
Yes.
But wait, this introduces a severe mathematical risk, doesn't it?
It does.
Like, how do we know the AI isn't just being lazy? What stops these new de novo nodes from just silently learning the exact same biological pathways that are already locked in the frozen nodes?
That is the exact problem. How do we ensure they're actually finding something new and not just duplicating the old map to lower the error rate?
Yeah, how do they do that?
To enforce this, the researchers apply something called the Hilbert-Schmidt Independence Criterion, or HSIC for short.
Okay, that is a very heavy mathematical term. A, quote, kernel-based measure of statistical independence. Before we even talk about what it does, what is a kernel in this context?
Sure. So a kernel is essentially a mathematical function. It takes complex data and maps it into a much, much higher dimensional space to make it easier to find hidden patterns or boundaries.
We've got higher dimensions.
Right. And the HSIC uses these kernels to constantly compare the behavior of the new nodes against the behavior of all the frozen old nodes. It is aggressively looking for statistical independence.
So it's not just like checking a list of genes to see if there are duplicates. It's looking at the overall behavioral pattern.
Yes.
It's almost like looking at the shadow cast by an object.
Yeah.
If the new node's shadow moves in the exact same pattern as the old node's shadow, they aren't actually independent. The HSIC ensures we are looking at a completely different orthogonal angle of the biology.
That shadow analogy is perfect. If the HSIC detects that you can predict the activity of the new nodes simply by watching the old nodes, it applies a massive penalty to the AI's loss function.
Another tax.
Another tax. It literally makes it mathematically painful for the AI to overlap. This rigorous statistical check guarantees that our new de novo programs are purely capturing the novel disease effects, completely disentangled from the baseline healthy biology.
Okay, we have spent a lot of time on computational theory so far. You know, the linear decoders, L1 budgets, architectural surgery, and the HSIC kernel mapping. Let's really ground this. Does this math actually hold up against the messy, totally unpredictable nature of a real human immune system?
It really does. And the researchers ran several real-world case studies to prove it. The first one involved mapping peripheral blood mononuclear cells, PBMCs, from patients with lupus.
Okay, lupus.
Right. They took these immune cells, left some untreated as a control, and stimulated others with IF and beta, which is a very potent cytokine that triggers a really intense immune response.
Honestly, this sounds like a brutal test for an AI. You got the technical noise of different patients, the baseline disease of lupus, and then on top of that, a massive chemical perturbation for the IF and beta.
It's an incredibly noisy data set.
And the paper notes that when they ran this exact data through a traditional gene set enrichment pipeline, specifically an algorithm called Limifry, it completely failed to provide useful insights.
Why do traditional methods just faceplant here?
Well, traditional methods like Limifry, they often really struggle with complex, nonlinear batch effects across massive atlases. They essentially look at average gene expressions across populations. Because the technical noise was so loud in this specific data set, the traditional algorithm could only detect the broadest possible signals. It spit out incredibly generic terms like immune response.
Which is totally useless. That's like going to a mechanic with a broken transmission and the diagnostic tool just says, car is broken.
Exactly. It doesn't tell you what to fix. But expiMap operates fundamentally differently. It performs end-to-end learning. It is actively solving for the batch effects and calculating the gene program activity simultaneously within the exact same network.
Oh, simultaneous calculation. Right. And because it cuts through the noise so effectively, in the B-cell populations, expiMap was able to identify highly specific, granular biological activities driving the reaction, like B-cell receptor signaling and antigen presentation.
It actually found the specific mechanism. But here is the critical benchmark for me. Did forcing the AI to be interpretable, forcing it to use those labels, ruin its ability to actually integrate the data? The paper compares expiMap against unconstrained pure black box models like SVI, Seurat v4, and Symphony.
And expiMap actually beat them.
Really?
Yes. It outperformed SBI in seamlessly blending the untreated query cells into the healthy reference map, while perfectly keeping the perturbed IFN beta cells distinctly separated. And it outperformed them because of the domain knowledge, not in spite of it.
Because it had the human labels.
Exactly. By explicitly knowing which genes belong to the IFN beta response pathway, expiMap could anchor the perturbed cells and separate them mathematically from the control state. The biology actively guided the math to a much better solution.
That is wild. And case study number 2 is even more fascinating to me. They used expiMap to analyze single-cell data from patients with severe COVID-19.
Right, and in the COVID-19 data, the model was actually able to identify specific, potentially druggable circuits that were hyperactive in severe patients. For instance, it isolated the RIG-IMDA5 pathway.
What does that pathway do?
It's a cellular circuit involved in recognizing viral RNA, and it happens to be a key driver of the cytokine storms that make severe COVID so dangerous.
But the detail in the paper that really stands out here involves the UAMP visualizations. For you listening, a UAMP is essentially a global 2D map. It compresses all those complex latent dimensions down onto a flat piece of paper so we can actually look at it visually.
Right.
And on the standard UAMP, the CD8 plus T cells from the COVID patients were sitting on a completely different side of the map than the IFN beta treated T cells from the lupus study. They look like completely unrelated biological states.
And if a researcher only looked at that global 2D projection, they would definitely conclude those cells have nothing in common. But expiMap doesn't just look at the global state, it looks at the individual interpretable latent dimensions.
The specific gene programs.
Exactly. When they interrogated the specific gene programs, expiMap revealed this hidden layer of shared biology. Despite their global differences on the map, both sets of T cells were firing on the exact same highly specific interferon signaling programs.
It found the shared signal completely hidden deep inside the noise. That's incredible. And we'll just briefly touch on the third case study. They moved completely out of the immune system and applied expiMap to pancreatic cells and type 2 diabetes models.
Right, and the framework successfully captured this specific loss of beta cell identity and the dedifferentiation process as the disease progressed.
Which really proves the framework is incredibly versatile. Across immunology, infectious disease, and endocrinology, expiMap consistently demonstrated that injecting human knowledge into deep learning does not hurt computational performance. In fact, it unlocks insights that pure black box models entirely miss.
It's a game changer.
Okay, here's your final mini recap of these clinical applications. First, simultaneous computation. By solving batch effects and biological mapping at the same time, expiMap provides granular diagnoses like B-cell receptor signaling, where older algorithms just guess. Second, better integration. Use up biological knowledge actually helps the AI map cells more accurately than pure mathematical models. And third, uncovering hidden links. expiMap can find shared specific pathways between diseases, even when the overall cells look completely different on a global scale.
Perfect.
So looking at the entire landscape of this paper, what is the ultimate takeaway here?
The takeaway is really a paradigm shift in how biological research will be conducted. Historically, computational biology has been a two-step process. First, you run a complex AI model to align the data and remove the noise. Then, you export that aligned data and run completely separate, often rudimentary statistical tests to try and guess what the AI actually did.
Right, and every step compounds the error.
Exactly. With domain-aware representations like expiMap, the alignment and the interpretation are the exact same mathematical event. We retain the raw world-class mapping power of deep neural networks, but we gain the ability to immediately look at a latent dimension and definitively say this specific gene pathway is driving this disease.
We finally have a translator for the map. And that brings me to a final thought I want to leave you, the listener, with today. We've talked about how expiMap's soft membership, that L1 regularization budget, allows the AI to pull in new genes. It is actively refining our databases based on raw, objective data. It isn't just reading the human encyclopedia, it is actively correcting our typos. It's finding new genes for old pathways. And with architectural surgery, it is learning entirely novel de novo disease mechanisms that humans haven't even discovered yet. So here is the question to ponder. At what point does the AI transition from being just a high-tech reading glass we use to look at biological textbooks to becoming the primary author of those textbooks? If an explainable AI can learn novel biology completely unconstrained by human bias, what other complex fields are about to be rewritten? Are we going to see programmable, interpretable AI mapping the unwritten laws of quantum chemistry or astrophysics, outputting discoveries not in abstract math, but in human concepts we can immediately understand? So here's the question to ponder. The map of biology has been redrawn, and for the first time, the AI is writing the labels in a language we can finally read.
The implications are honestly profound.
Thanks for taking this deep dive with us. Keep questioning the map.